So welcome everyone to this week's presentation at the Science Circle. We are doing a kind of last minute preparative thing because our scheduled speaker was not able to make it. And so of all the weeks to end up having this issue, this is like one of the best ones because it's an exciting time in science because the Nobel Prizes are announced. The winners are called at least sometimes very early in the morning. They have the press conferences. And then what's also particularly nice, and this leads into my talk, the Nobel Prize webpage compiles a huge amount of that's the Zoom link, compiles a ready to digest and understand amount of information with nice illustrations on their webpage. So I'm putting that link in local chat, and you'll see that that's where pretty much my illustrations and slides are derived from those. And this week was actually also particularly interesting for me in that two of the three prizes, both medicine and chemistry, actually intersect with my professional trajectory as well. And so these are two topics where I have a particular amount of insight or conversation that we can have. But what I'm really hoping for today is that, one, I just want to make sure I convey what are the kind of bottom line key aspects and takeaways of what was awarded, and then try and get into as quickly what the implications of those are that usually does interact with society and people in general. In fact, all three of these prizes are great examples of that as well. And so to introduce myself officially, I'm Stephen Gasior. I am a molecular biologist by trade, I have a Ph.D. I also used to teach college biology for about 9 years. And so let's get into it. Medicine, Victor Ambrose and Gary Ruvkin, for the discovery of microRNA and its role in post-transcriptional gene regulation. So again, just from the get-go of what it was awarded for is something that is a basic understanding of how cells and genes and expression and all these things work. That was a paradigm shift from what was previously known. And as you can imagine, there can be some very interesting immediate pharmacological impacts for this. This comes on the heels of last year. The award was for the development of basically the ability to express mRNA within cells. And so this whole concept of this RNA intermediate, RNA doesn't just carry information. It's just not, oh, we have DNA. That becomes messenger RNA. And then that messenger RNA becomes a useful carrier of protein information. That RNA is this very versatile molecule that does lots of different things in the cell. And so their overview that the Nobel Prize Committee provided, what it's demonstrating here at the top is just this reminder of how we think of the paradigm, the central dogma of molecular biology, which, like I've said before, that central dogma has been contradicted and shown to be plenty of exceptions many, many ways over many, many times. But you start from DNA, and that we think of as the functional gene that becomes transcribed into messenger RNA, and then translated into a different type of molecule into protein. And then these proteins are the workhorses of the cell. They are part of our structures. They are the catalyst, the enzymes that make things change, important for almost everything. And that what you can have in different cases, the regulation of this, and what they're showing is that depending on the cell type, it's the level of expression usually that tells you where and when a protein is being expressed. So the work that they're awarding is this interesting aspect of certain types of RNA molecules. So again, one of the honorary awardees is the nematode C. elegans, which again doesn't get its own medal, don't get its own little box anywhere. But what was observed was that there was a period of time where when C. elegans was first suggested by Sidney Brenner to be a real workhorse model organism, that people just started making mutants. They just tried to mutagenize them and see what happened with the worms. And there was this pair of mutants that seemed to have opposite effects. There's something known as Lin4 and Lin14. And these are things that changed basically certain stages of development where cells are being turned over or rapidly expanded. And they seem to have opposite effects. And so the laureates worked on these to demonstrate that these do seem to be in some way complementary to each other, that mutations in one, and when you combine these together, you get interesting effects. And that in fact, if you find mutations in one, they can basically get rid of the mutations that cause the effect in the first place. And so there's some sort of interaction that was going on here. And when they went to actually clone the Lin4 gene, this was the first example when they found by cloning that it's like, hey, wait a second, there's no protein being made here. This RNA, this is too small to make a protein. And so the other thing that they noticed, and this is diagrammed a bit here in that box on the right, is that there were stretches of the Lin14 mRNA that were complementary to the microRNA sequence. Now, when I say complementary, when you look at the red paired up with the blue, you see some black vertical hash marks. And this is a reminder that DNA works in pairs, that you have A that will bind to U, C will bind to G, and U will bind to A. And so you see this stretching along there. So again, U is uracil, and so we think of DNA where it's A, T, G, C. Those T's get converted to U's. And that's what they demonstrated, is that when this Lin4 is being expressed, is that it's in some way is binding to and finding the Lin14 RNA, and what they demonstrated was that it limited the amount of protein that got expressed. And so that is, in a nutshell, the key aspect of what these microRNAs do, is that they're these small little genes that are complementary to one or even several examples of other protein encoding genes in the cell, and they modulate their expression, usually by reducing the overall amount. And so one thing to keep in mind is that this is a very powerful technique that in many ways is, again, using examples of how RNA works. Now, one thing, RNA doesn't do this by itself, is that this is showing a reduced and simplified interaction here, but there are very specific proteins in the cell that then come in and modulate this binding, actually are the activators for how protein expression is decreased. Now, a follow-up discovery to this is that another microRNA that was subsequently cloned in nematode, LET7, was then demonstrated to be prevalent all across eukaryotes. Well, I should say in particular multicellular eukaryotes. That if you go back, 700 million years in ancestral evolution, that these RNAs, these microRNAs, are doing stuff that are modulating expression. And then on top of that, they found thousands. So the human genome has thousands of these small microRNA genes that work in different tissue-specific patterns that actually help modulate expression. If you mutate some of these become, in many cases, big developmental problems. However, they've been found to be important for all sorts of tissue specificity, involved in some cases you can find cancer, those other diseases as well. So this is kind of a summary from the Nobel Prize page showing this. And so I think this is one really interesting highlight thing that is in contrast to what I'll talk about in the next slide, which is that this microRNA mechanism is something that seems to be very specific to multicellular organisms. That this is not, well, there's a parallel to what's happening in, say, prokaryotes. This is something highly unique to what we see in eukaryotes. So I do want to do a quick flashback because in 2006, the award for physiology or medicine was awarded for something known as RNA interference. And so RNA interference, the award to Andrew Fire and Craig Mello, and again, honorary nominee, the nematode, C. elegans, is that the box on the right-hand slide is giving just a quick breakdown of what RNA interference does, which is these small RNAs are made when you have complementary stretches of RNA, and these really small ones go into a complex called dicer, and the RNA interference silencing complex RISC protein then goes around and finds other complementary sequences and just degrades that RNA. And so the power of this, again, is something that modulates gene expression, although in many cases people think the forerunner of where this actually came from, this mechanism, was that it was a very powerful way to be a bacterial immune response. That if you're a bacterium and you're being invaded by a virus, then the ability to stop that expression in its tracks, specifically to the virus, is maybe the place where this developed. But I think the deep evolutionary roots of this are still debated. This is, so I saw in the chat somebody talking about serendipitous awards and prizes and how serendipity and the recognition of what's going on can lead to Nobel Prizes. Is that RNA interference was actually discovered accidentally when people were trying to make flowers more red. They said, oh, we know what gene makes red, and so if we try and express more and more of it within the plant, then we should get petals that are more red. But then what they found in some cases was that extra expression of that gene made it less red. And that's because, in fact, it was making these double-stranded RNAs going the other direction and actually interfering with the expression of the endogenous gene and setting up this RNA interference. So I think that, in this case, like tagline is reminding us that Pasteur once said that discovery favors the prepared observer, and again, if you have the time, dedication, and smarts to follow up on it. So, the implication of medicine, and this, again, I'm mentioning the RNA interference because we think about lots of ways that RNA interference has been thought of as a way to do a drug delivery, a pharmaceutical therapeutic. Although in both cases, we're really understanding some sort of biological process at a fundamental level. What does it mean to be a cell, and how do you become a cell, and how do genes become effectors of cellular activity, etc. And so this was, again, a major overturning of this. And a reminder that when we think about the term gene, that, and this is something that's been around, I think, for a long time, that we tend to group genes into things that make proteins. But we have to remember that genes, at least in my definition, are effector elements within the cell that have a phenotype associated with them, have an action and activity. And so I think even these small genes are a reminder that we have to be careful about how we define things. And then it's an amazing lab tool for controlling expression. So that's one of the things that has been used. You can buy kits that are microRNA kits to do things that help you understand how cells work. It's a great way to turn off expression of a gene and then analyze what happens. And that's, the basic paradigm of molecular biology. But of course, it's very useful in plant and human biotechnology. One thing, I actually listened to the press release conference for this, and I feel like the Nobel Prize Committee kind of missed out on the fact that the concept of using small interfering RNAs to interfere with translation, that actually is something already in therapeutic trials. They're not exactly the microRNA full variant, where again, there's a larger RNA being made before you get down to the actual effector microRNA. So maybe that's what they were referring to. But they also were very human-centric, and there's a company out there known as Tropic Biosciences. And what they're doing, and what I captured from their YouTube video, is this technology can be used in combination with CRISPR, where if you modify that sequence of an RNA that's in the cell, you can retarget it. You can say, wow, okay, it normally targets this gene, or maybe it doesn't target many genes at all. Maybe it's an evolutionary remnant, but you can reprogram it to select a new target. And if the downregulation of that new target can help you with yield or robustness of the plants, or in some cases, you can use it to target a pathogen like like a virus or even a fungus. And so this is a company that actually is already announcing and talking about products. And yeah, Roy Sutton mentions we can save the banana, and that's actually one of the target plants for Tropic Biosciences, is thinking about the plants. Now, let me just quickly mention the company I work for recently announced a collaboration with this company, and so that's why it was on the top of my mind, and I'm familiar with the company, although I met them at a conference many years ago as well. But I don't know what's going on with that. I don't know all the plans. I'm not involved in the particulars of it, but just let me say that there is, I'm mentioning a company that does have let's call it an entanglement with the company that I work for, although not something I'm involved with. So yeah, no, this is very powerful. So I think, again, the Nobel Prize Committee they may not be, this is a very niche thing that I'm not blaming them for not being aware of it, but something that in agriculture, in combination with I think somebody mentioned the Doudna Charpentier 2021 Nobel Prize for CRISPR, although that was in chemistry, was awarded for. Thanks, Mike, 2020. All right. So again, there's a couple of questions, and we can talk about some more of the impacts maybe a little bit at the end. Is that you want to think about these as therapeutics, that once you have a basic understanding of cellular processes, and then you can start targeting very specific things as a way to remediate the problems that are happening. So somebody mentioned aging. And I think that's the main one. That, again, these are definitely ways of thinking about doing that. Although I will say that in many ways, the biotechnology world is a little bit more excited about CRISPR as a therapeutic intervention right now. All right, let's talk physics. So John Hopfield and Jeffrey Hinton for foundational discoveries and inventions that enable machine learning with artificial neural networks. Now, again, this is the one I had the least background in, but in reading through the scientific background that the Nobel Prize Committee provided, what's actually interesting is and this is why I think the lens of science is really interesting to understand, is that a lot of the basic principles of how we mathematically and try and model the way the brain works, right? We have a natural machine, the brain. We know it has neurons. We discovered things about how neurons work. But even in the 1940s, people were trying to understand, how does thinking work from a mathematical perspective? And one of the key things about neurons is that neurons, the basic way of thinking about them is that they're on or they're off. That neurons represent a binary state, which you can think of as 0 or 1, although in that case it would be they are either polarized or they are depolarized. And that, of course, we're talking about 9 billion neurons in the brain, that the combination of how a particular set of these nerve propagation events are occurring that in some way is recognizing states of information. And then also those states of information can be turned into an action. And so one of the things that they were modeling in terms of memory and recognition and pattern recognition is that some state of a bunch of nodes of neurons that are on or off during a nerve propagation event, basically creates that information state. And so what we have on the right was this idea and work saying that, again, modeled in the 1940s and 50s, is that you can think of a node network as something that represents these events. And so that's where Hinfield started making progress in terms of actually being able to computationally model and create these things. Now, one thing that I want to mention first is this theoretical background behind how you can computationally develop the right answer, or at least the best answer for some pattern recognition. And so what I'm showing here is this idea that you have a landscape, and this is very similar to what we think about in thermodynamics or even in protein folding is a good example, or protein variation, is that on an X and Y axis, you can represent some particular state, and then the Z axis, thing going vertical, is what is the energy of that state. And if you recall from physics, what we like to be able to do, or what happens in the universe, is that things go to the lowest energy state. That if you can in some way create a lowest energy state, that matches the pattern you're looking for. And so that was one of the major breakthroughs behind this, is that as you model learning, if you can create a set of nodes, a node state, the combination of node states that leads to a network node state that represents this low energy, then that's your pattern. And so that's what they're showing here on the right, is that, you're given an input, and it kind of looks, and it's supposed to be a letter, right? There's something where you're looking for a letter pattern. And then within all the 26 letters of the alphabet, let's just say they're all capitalized, that suddenly this one is recognized as corresponding most closely in terms of a low energy state to a J, and that's better than it would look like compared to an A or a K or a Z. And so, I really like this explanation. I think it works pretty well. And, these thermodynamic states are something, or these energy landscapes are something I'm very familiar with from protein engineering. So I hope that's enough of an understanding. I mean, the basic point is that you can basically try and find some sort of node state that recognizes some pre-matched pattern. And so this Hopfield associative network, for which he's been awarded the Nobel Prize, is just this generalized concept that within a computer algorithm, using certain principles from understanding spin states and the energy of atoms or the subatomic particles, that basically this thing known as statistical physics is a way that then one can start to translate this into a computer code that can be useful. Now, the next set of developments within this to where it's useful is what's called a Boltzmann machine. And the main advantage of a Boltzmann machine, instead of having a whole bunch of active nodes and just understanding that state, which becomes super computationally intensive. So tagline, I think, let me just, Roy mentioned that in the artificial neural network, each node has a function like y equals 2x, and they train the coefficient like that too in training phase. That if you have to do this and understand the math for each node, then you have this geometric progression of how much data you're analyzing every time you add a node. And so that's where the computational power became very limited to do this. And so two things happened. Is that one, in terms of refining the data processing and understanding how to make this useful, was this Boltzmann machine, where we think about hidden nodes that are important for data processing and can guide the processing of the data, but are not necessarily registered within the data that's the final output. or within the pattern recognition. And so this was very, very useful. And then the restricted Boltzmann is an example where, again, not every node is connected to every other node. And so now you can have a process. Now you have to be conscious of your process flow, that since you're restricting the total amount of data states, you're actually, in some ways, creating less power and less states that you can work with. But at least for a particular machine learning task, then I think you can create something that's very powerful with much less nodes and thus much less computational power. And that's actually the other thing that Hinton was recognized specifically for is, one, the basic idea that information can be stochastic. So there's a term that I hope this is the right application of the term, but something known as fuzzy math, fuzzy like peach fuzz, that as long as you have an answer that approximates what you're working with, then within a large data set, that's fine. And one thing that's powerful about that is instead of having to exactly model every single computational number, if you can use less computational power for fuzzy math, but it's highly approximating the actual answer you want, then that's a great way to get that to work. I hope that's a good explanation. I'm not a mathematics or computational expert. So what are the implications of this is basically this whole power of understanding data processing in a whole new way. And that this pattern matching can be used for a variety of fields because something known as deep learning can be enabled. And so in many ways, what this is that you can actually create outputs that are based on, it's going to be similar to a bunch of stuff and information I was trained on, even though it's not exact. I don't need exact data to provide you an exact answer, but I can have enough similar data to provide an approximation of almost certainly what is the best answer. And so you might remember that DeepMind was a company that was very involved in trying to help with medical diagnoses, as well as create machines that can basically beat every human brain in the world at chess. And again, the way a lot of chess programs were then made is they just let these algorithms play against each other. And just going through all the computational space, they learned all the best ways to actually do this based on the rules of how the game's supposed to be won. A couple highlights of how this technology has been used. The Higgs boson detection was an artificial neural network highlight. Again, they were looking for very specific energy signatures in a gigantic, vast amount of data. And so trying to recognize those patterns in that giant swath was, of course, something that almost tailored designed for artificial neural networks to do. Another one that the Nobel Prize Committee highlighted is mammary tumor detection, or again, they just said in general, medical diagnostics. But here's a really interesting example that, doctors themselves, in terms of recognizing whether a particular pattern in an X-ray from a mammogram really represented cancer, or maybe just some other sort of artifact of the light waves, or some normal regular structure that's just rare in some people, that excuse me for a second. Okay, so that if you have enough training data where you have, given the advantage of additional time, that is to say, here's someone that was given a mammogram, and then 10 years later we know that they were definitively diagnosed with it. So your data, your training data set is given the advantage of absolutism in terms of whether it represented a positive or a negative tumor, then you basically can use this really good training data set where the computers get very good at understanding the subtlety and can do that pattern recognition of what is going to be a tumor versus what's not a tumor. And I think all this conversation we've had in the past 4 years about artificial intelligence, the ability to train data and create this pattern recognition, this is the major enabling steps for AI. Now, in the interview with Hinton, he talked something about the backtrace algorithm. That backtracing is one of the key features to this next step of generative AI, and that was a key part of this, although I think that may ultimately be recognized as a separate stage or something separate for the Nobel Prize Committee to award. Because I do think the generative AI algorithms are something that are not just pattern recognition, although it's enabled by pattern recognition, it's also a synthetic synthesis of steps that are rather interesting. Ariana kind of mentions Big Blue can be Big Brother by AI. And in fact, when Hinton was interviewed by the Nobel Committee for the press release announcement, he hearkened back to some interview he did a few years ago, where he was mentioning that, one of the real possibilities of this technology is the destruction of mankind. That we could end up ultimately having an artificial intelligence that views us as competition, and that we've enabled in too many ways to do that. And of course, that's a wonderful place for science fiction writing, is this idea. And so I think the Nobel Prize Committee, of course, decided to highlight a lot more the use of diagnostics and the positive uses of AI, as well as many of the scientific uses. And that last scientific use they highlighted in a very particular way, which was to talk about another Nobel Prize in chemistry. So AlphaFold, which is something I'll get into a little bit more, is a program for predicting protein structure based on the deep learning of previously solved protein structures. And you can use it to thus make a pretty good guess at what some novel protein's structure may be. And so, with that said, well, actually, wait, any sort of conversation or discussion that people want to have about the physics prize? Brini is mentioning that Hinton's at some point said there's a chance, I hope you mean like recently, that within the next 5 to 20 years, AI may try to take over. It's interesting coming from him. Some of my thoughts on it, maybe we can go into with some more discussion at the end. I don't think we're in quite a Westworld type scenario, even within 20 years. Oh, the Canadian, YouTube recommended me that, I think, something from Canadian TV. So Suzuki also mentions kind of an interesting thing from Bing that, again, I think it was a New York Times journalist, that the AI wanted to be his mistress and run off with him. And I think, this is one of the things, too, is that, at least from my point of view, when we think about these generative AIs, is that when you give something the power to do something, it's doing it in a certain way that doesn't necessarily imply consciousness or intent or a viewpoint, right? And so it mimics something that when we interact with each other, we say, oh, this person has this motivation, or they're thinking this, because we know that they are capable of those things. But that you can't necessarily take that interpretation and layering of intent on conversation just because you've seen intelligent conversation. So, anyway. Oh, so Tagline mentioned something about using AI to screen for tumors and that false negatives would be a problem. Well, and whereas false positives would be safer in practice. I think the thing that people have shown over time, and Karthik is actually an expert on this, so he did his graduate work on that, is that in both cases, you reduce the number of false positives and false negatives over time. Although you do mention something that's actually really important to point out, is that there probably are lots of cases where we'll treat a tumor something we recognize to be a breast tumor are things that are being detected that were never detected before, right? And we don't necessarily know that in all those cases, the outcome of just having a tumor becomes a life-threatening tumor, because you could then uncover and discover the idea that there are lots of benign tumors that exist that in fact the person never would have come to the hospital to be treated for because there were no phenotypic or symptomatic outcomes or aspects of it. And so I think there's a very subtle way of thinking about how once you get into large data sets, what sort of ascertainment biases can come into play. And so people have argued that we've probably overtreated cancer, mammary tumors, for this reason that we're getting better at detecting them, and then we always assume that they're going to become something worse. Although the flip side of that, of course, is we now diagnose tumors using a lot of amazing biology, because we can take a biopsy, we can actually do a transcriptome or metabolomic analyses, again, also aided by AI in some cases, to understand what transcription state may represent a future tumor versus not. So, okay, I'm going to address this comment from Roy, and then we'll move on to the chemistry of, one of the things that AI, once you turn it into something that's also a chatbot, something that can then put information or chat into something that can be reacted to, is disinformation and made up stuff, and that you can enable these at a much larger scale, the economy of scale with bots and AI that can influence people to make erroneous decisions. And I think that is, we're seeing that play out, right? We already know that we're seeing a lot of that play out in many cases around the world, both for economic benefit as well as political influence. So again, it's certainly fair to us. I think it's certainly fair of Hinton to describe that there are negative consequences to these discoveries. But as we get into, I think, the chemistry, we can definitely focus on a real great advantage of these. So in chemistry, the Nobel Prize was awarded to David Baker, one half the prize, for the discovery of computational protein design, and then the other half shared jointly by Demis Hassabis and John Jumper for protein structure prediction. And so the next slide is a reminder a little bit about protein structure and how that relates to things, is that within biology, there are, not including things with modifications, there are 20 amino acids, and that these 20 amino acids have different types of chemistry associated with them, but they're all connectable. They're like Legos. You can connect them up to make a string, and then you get a protein that then forms some sort of structure. And it's the structure, the interaction of the structure to itself, as well as this interaction with other things, that are important for what it does. And so here's a little bit of a reminder. Some of the layering of that is that you have the primary structure, that's the sequence, and then secondary structures, which are localized interactions for proteins that form certain shapes. And then tertiary are those shapes interacting with each other. And then quaternary are where completely different peptides, completely different stretches of primary sequences come and interact with each other. And so what was known since, I want to say 1973, the Nobel Prize in Chemistry was for the demonstration that RNase, that you could unfold it and it would naturally fold back into an active structure again. Now, I think that's a rather outlier of a case when it comes to protein structure function overall. But it was a demonstration that this primary sequence you see here is the primary and only determinant of what structure it ultimately forms. Now, in many ways that's wrong, but functionally let's just work with that as being the way we think about it. And certainly true enough to be something that we can definitely be a foundation for further study. And so AlphaFold, in particular the variant AlphaFold, let me go back. So one of the golden gold rings on the carousel was for us, once we had enough computational power, could we take a primary sequence and then predict the final structure? And there actually was a competition set up called CAPS, and I can't remember what that acronym stands for, but a yearly competition for people to come and compete and show that their program can correctly predict structures. And that competition actually came to an end because the AlphaFold 2, which again is a product of DeepMind, which is from the Nobel laureates. The combination of the awardees was for having set up this company and then also help refine this technique. And then, Sis, remind me to answer that question just a little bit. Let me go through what the award was for first. That AlphaFold, for the first time, was getting very close to this goal line of 90% of the time accurately predicting the structure of a protein. They actually came to with 87% in that year's competition, and it's almost certainly gotten better since then. But the way this works, and this comes back to this whole deep learning idea, is that AlphaFold's trick was to take all the known structures, but then to also do a comparison across every version of that protein, even across species, and to see where those different amino acids could be substituted or not substituted, where that impacted function, where it didn't impact function, and then that helped it build this landscape of what is the most likely way that a similar sequence would fold. And so that was kind of their special trick. But now AlphaFold is successfully used to accurately predict protein function. And so that's where the AI comes in, and through several rounds of iterative analysis can take a primary sequence, and then in many cases, as long as you have a good foundation of that protein structure in databases, can actually do a pretty good job of predicting a novel protein. So any questions on the basic aspect of how that works? I mean, it really the basic idea that you need to come away with is that they realized a type of algorithm that can say, hey, give me a primary sequence, I can predict what it's going to look like as a structure based on all these databases that have structures, and then a little bit of special algorithmic sauce. Okay, so now in practice, what's happened with AlphaFold, and this comes back to the quaternary structures, is that AlphaFold 2 itself, when we think about that program, it's predicting one peptide at a time. And so it's saying you input a peptide, and that spits out what it thinks it will fold into. And again, one thing that's important, it gives you some degree of confidence for how well it thinks it predicted it. There are variants of AlphaFold, and you have to find these get new names because people do different things, that once you sit down and try and understand a protein-protein interaction between two different proteins, or what are called small peptides that may interact, or ligand receptor interactions, once you have two different proteins, these use some of the principles of AlphaFold learning, but they're not, we wouldn't specifically call them AlphaFold. So, sorry, let me go back. I see there's a, quinary structure. Yeah, actually, so that's not a term that I think, okay, that's not a term I've come across, but from what I understand of it, which is that, and this is kind of going back to my original point that I made earlier, is that if you think about these proteins, and this is the limitation of this data, and that's, to be very clear in practice, the limitation is the database for the deep learning is a static version of a crystal of that protein. And that does not represent what that protein is doing in the cell. It doesn't represent the amount of water. It doesn't represent the pH, the charge, in other words. It also doesn't represent another thing that's usually overlooked, which is that when proteins are being translated, that the thermodynamic state, this lower thermodynamic energy for the whole protein, may not be the same as just having the first quarter of it, the first third of it translated while it's waiting for the rest of it to be made. And so you can have there are a few examples where a protein folds the first half of it, and that's independent of the thermodynamic state of what happens with the second half of it. And so I think that that's an important consideration. And then the other thing that's important are chaperones. So they're these small proteins that recognize unstructured proteins in the cell, and that some of them are helpful and important for actually creating that final folded version that's in the cell. And so I think in many ways, this is a Nobel Prize that awards a paradigm shift that will also, many decades down the line, probably award another Nobel Prize for paradigm shifting this paradigm, because people will start figuring out ways to actually analyze the living state of proteins actually in the cell. Now, that's not to take away from the fact that this is super powerful and useful when you're in the lab. And that's where I want to get to one example next. For David Baker, is that he developed a program called Rosetta. So he was involved in this prediction competition, and he did pretty well several years, but he didn't win. But in the process of doing this, he actually realized, wait, could this computational power be used to have a program, you tell a program, this is what structure I want, and then it figure out what sequence you need to make that. And that sequence can be just based on the principles that you know of what the chemistry of the amino acids are. Does that make sense to everyone? That instead of trying to figure out the structure based on the sequence, now you're asking, you want the structure, figure out the sequence that would make it. And that's what Rosetta does. And so the Nobel Prize mentioned a few examples here. Basically, the linking together of small proteins into a large nanomaterial. Mentioned one that they developed one where you can use it as an effector, a sensor for fentanyl, which would be helpful for overdosing. Nanoparticles that can bind up an influenza virus, and that can be used as a way to interfere with viral function. You can think of something being a molecular rotor. So we have known rotors in biology. They've actually successfully made ones that are designed. And then other types of sensors based on, again, multimers interacting with each other. And so I think that's the really powerful thing. So I've been reading up on this technology recently, because I work in a protein engineering group within the company. And so we're very interested in these technologies. Now, what I think is interesting here, you see this in these examples, the overall size and complexity of the designed de novo proteins are actually kind of small. And you really don't see something more than 2 different types of proteins at a time being made to interact with each other. So again, give it a matter of time where we can develop more complicated things. But right now, I would say that the limitation of this is still some smaller things and smaller use cases for them. Let's see. So implications of this work, and I think that this is something that people have already been hitting on in the local chat, that from a fundamental understanding of biological processes and chemistry, this accelerates the basic understanding of protein structure at a so much lower cost. And so what it enables within the research space, or again, whatever sort of technology engineering space you want to talk about, is to more quickly have a basis for how you want to modify it or adapt it to some particular, say, therapeutic or other industrial purpose. And so that is something that is, extremely powerful for what we want to do for humankind, right? Now, specifically, the types of things that we think about within the industry is that when you have a category of proteins that you're already working with that has some sort of function, right? There's some sort of invention-driven process where you want to recreate it or make it better, then having these structures even if they're just best estimates and guesses, it's something where the human brain can then come in and either do library engineering or directed rational engineering to do that next step. So the company I work for, of course, is very interested in proteins that are known to kill insects. And we use these, the agricultural industry uses these as a way to manage pests within crops. And so we have lots of examples of this. And so using this to understand a novel version's interaction with the insect or how we can engineer it to make it work better, these are things that we were definitely invested in, very interested in doing. Like I mentioned in the examples, drug delivery or detection. One thing you can do to help so there are cases where your de novo protein can be the drug, and that's, I think, some good examples are very similar to, like, when you think about modern pharmaceuticals, a lot of it has to do with binding proteins or variations on antibodies, and so this gives you a lot more ability to design or understand how those are working. But the other thing, too, is you can actually use proteins to be a delivery mechanism even for small molecules, right? Once you understand how you can encapsulate something using de novo or novel proteins, then that becomes yet again another delivery mechanism that we can use. And then another one that was kind of mentioned by the Nobel Prize Committee was thinking about like novel chemistry. And I talked about this when I talked about heroes of evolution, talking about Francis Arnold, that a lot of times you can develop some really novel chemistry just by trying to take what you have and then push it in a way to work on something novel or new. And so de novo protein design is another good example where you can, once you start understanding what the structure of a plastic is, how some proteins might interact with that plastic, then you can start using de novo design to maybe do something that has a useful function like degrading plastics. And so that's kind of the space we're in. And so that's it for what I wanted to talk about. This is the roundup, some of the implications, the basic science behind them. And so I think I hope you enjoyed it, and let's discuss a little bit, because I hope you're excited about it as much as I was.